Hello, my dear lovely students. So, hope you all are in best of your health. Today, I'll be giving you a summary of the chapter Biomolecules. This whole chapter we'll be looking at in a very short way. That means we'll be looking at each and every concept. Now, first of all, let's talk about the biomolecules. The chapter headline is biomolecule. That means we will be talking about those molecules which are present inside living organism. So, let's have a look. So, biomolecules we will be discussing today, whole chapter summary basically I'm going to give you. So, to talk about what actually these biomolecules are are organic or inorganic compounds, or maybe some molecules which are present inside the living organism or the living cells. These are termed as biomolecules. See, biomolecule, right? Living molecules which are present inside the living cell. So it includes everything. It includes some macromolecules also, such as large macromolecules such as your proteins, polysaccharide, the lipid, the nucleic acid, as well as the small micromolecules such as primary metabolites, secondary metabolites, and the natural products. Now, to talk about the proteins you all know they are made up of amino acid. A chain of amino acid, they join together, they fold itself into a secondary and the tertiary and the quaternary structure, and what they form is a protein. Similarly, we have as a polysaccharide. Polysaccharides are basically what? These are your carbohydrates, your complex carbohydrates, which act as a structural component as well as storage component as far as the living organisms they are considered. The lipids are kept under the macromolecules, but actually the size is comparatively less. Now, lipids are basically, these are the esters of fatty acid. They are the, for example, in this case we have cholesterol also, we have some derived lipids also. So they are complex substances made up of different things. So all of this will be included over here. Similarly, nucleic acid, for example, we have polynucleotide chain, that is your RNA and the DNA, that is also included in the macromolecules. Now, let's talk about the analysis. Here, this is a complete chart from the NCERT. Here, guys, we are talking about the element present in non-living and the living matter. Now, overall, the composition is somewhere or other similar. That means in the earth crust also you will find those molecules or the substances, basically the inorganic substances. Similarly, in the living cell also you're going to find everything. But composition, yes, somewhere or other it is going to vary. But overall, they will be present over, if we talk about the plant cell, in that plant cells also, the substances, they will be similar, those which are present in the earth crust. For example, your hydrogen, the carbon, the oxygen, nitrogen, sulfur, sodium, calcium, magnesium, and the silicon. You can see the earth crust, this is how they are present, and in case of human body they are also present. Clear? This is the overall composition. You know, no need to learn the percentage, in which percentage weight in which it is present. No need to learn that. Just you have to remember that they are present. Out of all these things, out of all these elements, if I talk about, whenever we see the hydrogen, this is carbon, this is oxygen, the nitrogen, and the sulfur. This forms a major component as far as your living body they are considered. To talk about the inorganic constituent, so inorganic constituents, they are present in different form in the living tissues. For example, the sodium, you will find sodium as Na positive, right? You will find potassium as this, right? The calcium will be present as calcium 2 positive, magnesium will be present like this, and water. Yes, to talk about the water is most abundant as far as the living organisms are considered. Most abundant, I'll write it down here, the most abundant molecule. Most abundant molecule as far as the living cells they are considered. About 70 to 90% the cell is having just water, and then we have is a protein. The compounds, other compounds, these inorganic substances, they combine together, they form other compounds also. For example, NaCl you'll find calcium carbonate, you'll find PO4 negative, the sulfur oxide, all these things they will be present, but this is how they are present. Now, moving on to the next, when we talk, your NCERT talks about the chemical composition. See, chemical composition. Chemical composition means we have different substances which is present inside the cell. But how can we analyze it? How will we get to know that they are present? Now for this a simple experiment can be done. Here guys, what you have to do is a very easy experiment. One can also do that in the laboratory condition. For example, suppose you have to take any cell. Let's say you got liver, right? Liver is what? We are going to get the tissue. Liver is tissue, right? It's an organ, like some tissue you got. For example, the liver. And either you can take an example of a leaf, right? Whatever you get, what you're going to do is you're going to grind this. And how you're going to grind this? In the pestle mortar. When you will grind this, you have to add some liquid thing also, because without adding liquid thing, you will not be able to grind it. How will you do that? By the addition of trichloroacetic acid. How will you do that? By the addition of trichloroacetic acid. So what is this trichloroacetic acid? That is Cl3CCOOH. Trichloroacetic acid. So right, clear? So here you all know this is an acetic acid, and acetic acid is combined with the 3 chloride ions, 3 chlorine. So that is the reason it is named as trichloroacetic acid. Now what you're going to do is you'll get a thick slurry. You can say a homogeneous mixture you're going to get. It will be like a thick slurry. Now what you're going to do is you're going to sieve this. Either you take a cheesecloth or the muslin cloth, you have to sieve it. So when you're going to sieve it, you'll find 2 different compounds. See guys, you have this beaker, and here guys you have kept the paper, or sorry, the tissue, whatever it is. So you're going to pour your substances over here. Some things will come, some liquid substance will come over here, some will be present above. So one which is present in the beaker, that will come in the beaker, is termed as a filtrate. So filtrate will be one that means it is acid soluble pool. So because acid is a liquid, acid will come down, right? And those substances which are acid soluble, they will also come down, and everything will be termed as your acid soluble. So in the acid soluble, what you're going to find is the micromolecules, small molecules. I'll be talking about it. Whereas over here, the above the paper, above this cheesecloth or the muslin cloth, you will find some things are present, that is termed as a retentate. Clear? Filtrate is clear, and above you're going to get the retentate. So retentate, they are acid insoluble. They will not be soluble in acid, and they are termed as your macromolecules. What do we call it? Macromolecules. Now listen very carefully. To talk about your macromolecules, so macromolecules, that means these are those whose size is very big. And where are you going to find it? You're going to find it in acid insoluble pool. So they are found in acid insoluble pool. They will be found in acid insoluble pool, first thing. Now the second point is macromolecule. That means their size will be bigger as compared to micromolecule. So their size is more than 800 Dalton. Dalton is a unit, right? More than 800 Dalton if size is there, that means we are talking about the macromolecule. So in this case, we have a various example. Here what you're going to find is, you will find protein, right? You will find the polysaccharide or the complex carbohydrate, polysaccharide, right? What else? You're going to find the nucleic acid, which I'm writing it as Na. Nucleic acid you're going to get. What else guys you will get other than nucleic acid? The lipid also. But one thing you should remember, lipid size is less than 800 Dalton. But still they are kept under the macromolecule category. Why? Why? Sorry guys. Sorry. Because when we talk about these macromolecules, that means they are bigger size. And when we get the lipid, they are like, they are globular structure. Because when a plasma membrane breaks down, just a second guys. So you know, the lipid bilayer, whenever it breaks down, it is present in the form of a tissue. It is present in the form of a you can say the fragments are there. So they are less than it because it is complex with other substances also. That is the reason they're kept under the macromolecule category. Clear? So macromolecule is clear. Now let's talk about your micromolecule. Now let's talk about the micromolecule. Micromolecule, so name itself indicate, that means their size will be smaller. Micromolecule, so their size is, so basically they are found in, first let's talk about the found in acid soluble pool, acid soluble pool. Now what else? Now their size is 8 to 800 Dalton. 8 to 800 Dalton. So what are you going to get over here? You will find nucleotides. So here I am writing nucleotides will be there. Nucleotide. What else you're going to get? The amino acid. Amino acid. You're going to get the monosaccharide, etc. Nothing very complex. Hope it is clear, buddies. Macromolecules and the micromolecules concept. Now, to talk about the average composition of cell. Now we have seen that all these substances, they are present. We have seen the major macromolecules, they are present inside the cell. Now the average composition of cell which your NCERT talks about is the water. So basically it is having the water. So this was many times asked in the NEET examination. So maximum in the cell we have is a water. Clear? That is 70 to 90%. Then we have is a protein, carbohydrates, the lipids, nucleic acids, and the ions are present, which is even less than sometime 1%. So this you have to remember, very important this for your NEET from your NEET point of view. Now let's talk about the ash analysis. So why ash analysis is being done? To talk about the inorganic substance. So we got to know about the organic substances. Let's say how can we get to know the inorganic substances. Inorganic substances. So inorganic substances are those substances which we can determine with the help of an ash analysis. So how can we do that? So buddies, you are going to take tissue, anything, tissue, leaf, whose inorganic analysis you have to do, right? What are inorganic substances they are present? How will you analyze it? You're going to do the ash analysis. For example, you're going to take the tissue and you're going to burn it. Burn, right? What will happen? Your carbon containing substances, they will go out in the form of carbon dioxide. They will go, because majorly what you're going to find is a carbon. So, and what about the water? Water will also evaporate. Water will also evaporate. So majorly what we are going to found is, see, I'll talk about it. See, here in the proteins, the carbohydrates, in the lipid also, the backbone is of a carbon. That we'll see later on. So carbon dioxide, the carbon will go away. So this is how the carbon will go away, because we have burnt it. And this is how we keep it. We keep the tissue over here, and with the help of a tongue we hold it, and then we are going to burn it. Whatever which is left behind, so left behind, that will be termed as ash. That is termed as ash. Now this ash will be analyzed. Now this ash, what we are going to do is analysis. So analysis will be done, and this analysis is important. Either we talk about the functional group. Suppose we have to know about the functional group with the help of ash analysis. Yes, we can easily do that. There are various methods in this that you learn in the chemistry. Now other than that, for the biology part also, you can easily do that. Clear? Like what all inorganic substances that are present in them. Now there are various ways by which you can do that. So for this, two different terms which you have to remember is, the first one is a wet weight. First one is the wet weight. Wet weight. So what is a wet weight? Wet weight, that means we are talking about, guys, initial weight. initial weight. So initial weight of a substance, that is termed as the wet weight. Clear? And the dry weight. Dry weight. So what is a dry weight? That means after removal of the carbon dioxide. That means when you are going to weigh of weight of ash. So that is termed as the dry weight. These are the two different words which you have to remember. Now, moving on to the next. There are some other metabolites also present in the inside living organisms. Basically, they are found in the plant cell. So they are termed as a secondary metabolite. Why the different terms are being given? Let's talk about it. First name is given as primary metabolite. You know about primary metabolite. It's just that I'm giving you an overview of these primary and the secondary metabolite. So primary and the secondary metabolite, because these terms I will be using very much. So primary metabolites are those metabolites which are important for survival. Which are important for survival. For example, we have carbohydrates, we have proteins, we have vitamins. Everything will be included over here. Carbohydrate, protein, your vitamins, lipids, all these things, they come under this category. Whereas when we talk about the secondary metabolites, secondary metabolites are those which provide some extra properties. These are those which provide some extra properties. Extra properties it is going to provide. So what are these extra properties? For example, the pigments are there, like carotenoids, like anthocyanin. We have alkaloids like morphine and the codeine. Yes, they are also used in various medicines, the morphine and the codeine. For example, the codeine is used in cough syrups. So some extra properties they are going to provide. They are the secondary metabolites. Terpenoid monotremes and diterpenes. We have essential oil, for example, lemongrass oil. The toxin, arbutin and ricin. Lectins are there, drugs and polymeric substances. So these are the substances which are produced basically by the plants. And they provide extra properties. Now, moving on to the next, let's first of all move into the carbohydrates. So we are going to start this biomolecules, basically the macromolecules we have to discuss. So first of all, we are going to start with the carbohydrates. So what are these carbohydrates? Carbohydrates, they are what? They are termed as a hydrates of carbon. Hydrates of carbon. Hydrates, so they are having a chain of carbon, right? Their general formula is this CN, H2O, N, right? Or you can also write CN, H2N, ON. This is how, also one of the way by which you can write. They are defined as polyhydroxyaldoses. Aldoses, the name is given, they are hydrates of carbon. They are chains of carbon present. They can be having a aldehyde group. If they are having a aldehyde group, they are termed as aldoses. For example, glyceraldehyde and a glucose, they are aldoses. Or the polyhydroxyketosis. For example, they are having a ketone group. And you know the ketone group is present where? It is present internally. Now in this case, the example we have is dihydroxyacetone. Fructose in the other molecule that gives the structure on hydrolysis. Give these structure on hydrolysis. Carbohydrates are also called as saccharide because their components, they are the basic components of sugar. Now, here the two different words are given. The glucose, definitely we'll be looking at the structure. We have to know about the fructose structures also. But first of all, let's talk about the polyhydroxyacetone. What is the meaning of polyhydroxyacetone or the glyceraldehyde? Let's talk about it. Now, buddies, please have a look. Now, buddies, see. These are the hydrates of carbon. So on one side, if we have an aldehyde group, right? And here we have is just CH2OH. Because here it is written as hydrates of carbon, polyhydroxyaldoses. So here also what do we have is, see, we have a H, we have a OH over here. So this is termed as your glyceraldehyde. What is the name given? So name will be written as glyceraldehyde. Aldehyde. Glyceraldehyde. Glyceraldehyde. So this is also carbohydrate. That means the simplest carbohydrate. Here the 3 carbons are there, so the name is written as, so name can be written as trioses. Now, the second one we have is this one. 3 carbons are there. And if we have ketone group like this, here we have H, we have HO, and here we have H. Similarly, OH is there. We have one H. Here also we have an H. So this is also a kind of, you can say the carbohydrate. The simplest carbohydrates, the name is written as dihydroxyacetone. So what will be the name given to this? The dihydroxyacetone. 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 Clear, buddies? Dihydroxyacetone, and this is glyceraldehyde. So these are the simplest, and the common name is given as saccharides. Now moving on to the next, carbohydrates are generally classified as in sugar and the non-sugars. So we classify them into the sugar, like sugars are there, and the non-sugar. So basically sugars are what? Sugars, they are your monosaccharides and the oligosaccharides. Now why this name is being given? So monosaccharide, that means a single unit is present. Single unit, right? Single unit is present. For example, this is a normal sugar. For example, this is a structure of a glucose. If this is present singly, that is termed as the single sugar, the monosaccharide. Now when we talk about the oligosaccharide, that means 2 to 10 subunits are present. 2 to 10 subunits, if they are present, they will be termed as your oligosaccharides. Clear? Now monosaccharide, they can be aldoses and ketoses. Aldoses, that means they are having an aldehyde group. Ketoses, that means they are having a in-between ketone group present. Now if it is made up of 3 carbon, so that is trioses. If it is made up of 4 carbon, that is your tetroses. If it is made up of 5 carbon, pentoses. If it is made up of 6 carbons, this is a 6 carbon, we have is a hexoses. Similarly, ketoses, that means if they are having the ketone group present in between ketone groups, and it varies, the number of carbon if there varies. So accordingly the name has been given trioses, tetroses, pentoses, and hexoses. So hope this is clear, monosaccharide. Now let's talk about the disaccharides. So disaccharides are those which are disaccharides. So that means we are having 2 subunits are present. Clear? 2 subunits are present, and these 2 subunits, they are joined together with the help of one bond. This bond is termed as glycosidic bond. Glycosidic bond. In between glycosidic bond is present. That means 2 monosaccharides, they are joined together. Trisaccharides are those in which 3 monosaccharides, they are joined together. Right? So it is trisaccharides, they are somewhere or other like this. Right? Tetrasaccharides are what? Tetrasaccharides, that means 4 are joined together. So like this. And they are having the glycosidic bond present in them. And whenever they join together, hydrolysis occur. Right? That means water is being removed. Clear? So hope these are clear. So the oligosaccharides are clear. Now let's talk about the non-sugar polysaccharides. So non-sugar polysaccharides are those which are basically the structural element. They are the structural, that means they are important in the structure, or you can also say other one, that is they are the storage. So 2 functions are there. So accordingly 2 names are being given, the structure when they are present as a structure, or the storage. For example, when we talk about the homopolysaccharides, that means these are those which are made up of same type of subunit. So same subunits, they are repeated again and again. Same monosaccharides are repeated. Right? Same monosaccharides are there, and same monosaccharide, they are repeated again and again. Whereas when we talk about the heteropolysaccharide, that means many different type monosaccharide unit. Monosaccharide unit. Clear? So that means many monosaccharide units, they are joined together. When we have many monosaccharide units, they are joined together. So what they form is the hetero. Hetero means different. Hetero means different. Clear? Now, let's talk about the homopolysaccharide. In this case, the cellulose is there. Other examples, I'll write it here. Here I am writing the examples. For example, we have cellulose, we have a starch, we have glycogen, right? These example. In a heteropolysaccharide, we have an example like we are having hyaluronic acid. Hyaluronic acid, we have peptidoglycan, the heparin. I'll write it here. Hyaluronic acid, heparin, yeah, or peptidoglycan, clear? So all these are present carbohydrates, so this is how they will be classified. Clear? So we talked about the monosaccharide, we talked about the disaccharides, and we talked about this. Now oligosaccharides. In the oligosaccharides, so basically those disaccharides which are there in your syllabus, they are the sucrose, maltose and lactoses. So 3 examples, let's talk about it. Yes, so 3 examples are there as far as the oligosaccharides are considered. Here we have is a sucrose, we have is a maltose, and we have is a lactose. So sucroses, they are made up of alpha glucose and the beta glucose subunit. Here the glycosidic linkage which is present is glucose alpha 1 beta 2 fructose. Here we have 2 subunit, to alpha glucose and the alpha glucose, both of them are joined together, and the glucose alpha 1 and 4. That means in the first one, that is in the alpha glucose, first carbon attached to the 4th carbon. So this is how the name has been given. The lactose is alpha glucose and the beta glucose. The bond which is present is glucose alpha 1,4 glucose. Right? This is a glycosidic linkage. This is the type of glycosidic linkage which is present. That means the first alpha carbon is attached to the 4th one. First one is attached to the 4th one. First one is attached to the beta fructose structure of the second one. Now, polysaccharide, I told you the first polysaccharide which is there in your syllabus is your cellulose. Cellulose is the monomeric unit which is present in them, that is a glucose, that is a beta 1,4 linkage is present. Now, this is a most abundant polysaccharide. You know, they are cellulose is present where? Cellulose is present in the cell wall of the plant cell. It is formed by the beta glucose. It cannot be digested by the human, so we do not have enzyme for that, but can be digested by the ruminant. For this, the enzyme which is required, that is a cellulases. Celluloses, we do not have cellulose, so we cannot digest cellulose. Now, cellulosic paper is made up of the plant pulp and the cotton fiber. Now, second one we have is a starch. So starch is made up of D-glucose. You all know starch is what? Starch is a storage polysaccharide as far as the plants they are considered. Here they are made up of 2 different structure, amyloses. If they are present in a linear form, that is amyloses are present, and the linkage will be alpha 1,4. Amylopectin when they are present in a branched form. So when a linear structure is there, that is alpha 1,4, and when branching is there, the alpha 1,6 linkage will be present. Clear? Now, here one thing you have to remember is they hold iodine molecules, so that is the reason it gives a positive iodine test. If we talk about this, is this branched? Yes, guys, you have to remember that these structure, the starch is a branched structure. Now, next one we have is a glycogen. We all know about the glycogen. Glycogen is what? It is a storage molecule as far as the animals they are considered. Is it made up of glucose? Yes. This is also made up of glucose. Yes. If someone asks you it is a branch structure, you have to know, yes, it is a branch structure. Here the bond is present in such condition that they are not able to hold iodine molecule. That is the reason it gives a negative result. That means it is not going to give a bluish color. It is going to give a red color. So negative test it shows, and it has a branch structure also. It is also termed as, because it is a storage structure, whenever storage polysaccharide in plant is termed as, we call it as a starch. Whereas when we talk about the glycogen, it is in the human body, that is in the case of the animals. So that is the reason it is termed as an animal starch. Now next, another question, that is inulin. So this was asked in your NEET examination. Like what actually a inulin is, you should know it is made up of D-fructose beta 2,1. right? And it is a polymer of fructose. You have to remember, whenever a person goes for a glomerular filtration rate, it is related to the kidney. So inulin is being used over here. Now that is, you have to remember, it is a storage structure in the dialia, storage things in a dialia. Next we have is a chitin. Chitin, you know, the arthropod we have talked about in the animal kingdom. The chitin is basically a structural element that helps in the protection. It is also a homopolysaccharide. It is made up of NAG. NAG, you have to remember, see, both of them, they are termed equally. Chitin, you know, they are found in the arthropod as well as in fungi they are found. So we are talking about the arthropod, that means the animal, and nag in the Hindi, the nag is termed as a snake. So this is how you can learn things. So when we talk about the arthropod, this is made up of nag. So what is the full form of nag? That is the N-acetyl glucosamine. Nag in Hindi, that is it's a snake. So beta 1,4 linkage is present. Here the molecule which is here, the, you can say, the monomeric unit is NAG. Peptidoglycan is made up of NAG and NAMA, N-acetyl muramic acid and N-acetyl glucosamine. They are present, and they are present in the cell wall of bacteria. Now next one is a hyaluronic acid, that is termed as a cement. In the animal cell, the cement which is present, that is termed as hyaluronic acid. They are present in the cartilages, they are present in various connective tissue. Hyaluronic acid, animal cement. Here they are made up of D-glucose uronic acid as well as the NAG, that is N-acetyl glucosamine. Now moving on to the next part of this chapter, that is the lipids. Now lipids, you all know, many students, they get confused between, ma'am, what are lipids and what are the fats. So basically the fats are the type of lipid, we'll be looking at it. So lipids are what? Lipids, they are basically hydrophobic in nature. They are hydrophobic in nature. We all know about it, right? Hydrophobic in nature. And basically all the fats, all these lipids which are present in the body, mostly they are present in the form of a fatty acid chain. So this is how a fatty acid chain look like. This is also a long chain fatty acyl group. Long chain fatty acyl group is present. Now this is termed as a fatty acid. On one side you can see the acidic group are present. So in the body, the basic unit is a fatty acid chain, as well as most of the lipids they are considered. So depending upon the number of carbon atom it is present in them, a name has been given. Clear? Here guys, here we are talking about the 16 carbon atom, and 16 carbon atom, the name is given as a palmitic acid. Now arachidonic acid is also present, for example, which is having a total 20 carbon chain. Now this fatty acid chain which is present, now this is a linear chain, right? Are you getting this point? This is a linear chain is present. On one side you will find a acidic group, that is a COH group is present. Now let's look at this fatty acid chain. Now this fatty acid chain can be saturated, or they can be unsaturated. Accordingly, the different name has been given. So if they are saturated, that means they do not have double bond. No double bond. No double bond is there. Right? It is a linear chain. No double bond is present between the carbon and the carbon. Unsaturated, that means they have a double bond. They have double bond present. Clear? So saturated, that means no double bond. So what is going to happen is, whenever we are going to place them together, so they will place in tightly way. Like this is how they will be present. So this is how. So they remain solid at room temperature because they are tightly packed to each other. So they are solid at room temperature. For example, the ghee. Now unsaturated are those which are having the double bond. So when someone is having a double bond like this, suppose this is a double bond, so this double bond is going to create a kink, so with which they are not be able to tie it properly. So that is the reason they remain oil at room temperature, or they are liquid at room temperature. So accordingly name the saturated and unsaturated. Hope it is clear. Now another substance which is present in them, that is a glycerol. So glycerol is again alcohol. 3 carbon chain are there. 3 carbon compounds are there. Everyone is attached to the alcohol. Now the first type of lipid, you all know, that is the triglycerides. The triglycerides. So triglycerides, they are what? They are, see, this is our glycerol, and if glycerol, this particular glycerol is attached further to different chain of fatty acyl, different fatty acyl chain are present. If different fatty acyl chain are present like this, clear? So this will be termed as a triglyceride. So basic unit is glycerol and the fatty acid, and if they are present like this, they will be termed as a triglyceride. Three chain, fatty acyl chain, they are present on the glycerol. So this is a simplest form, triglyceride. This is also termed as a simple fat. This is termed as simple fat. So their number also varies. So definitely the name is given as R1, R2, R3. They can be 16 carbon, they can be 18 carbon, they can be saturated or unsaturated, depending on the type of glycerides we are, triglycerides we are talking about. Now the second type we have is a phospholipid. So phospholipids, they are what? Phospholipids, they are the derived. They are the derived lipid. Derived lipid. Here guys, what do we have is, we have the glycerol. On this glycerol, we have this R1 present, another R2 is present. At the third position, we have is this phosphate group present. Guys, can you see? So this is, the name is given as a phospholipid. And one example which was asked many times in the examination, the phospholipid, the example we have is the lecithin. Here you can say another R group is present. Clear? So this is termed as your phospholipid. Phospholipid, the another name is given as, what do we call it? The lecithin. One example in this case is a lecithin. So these are the compound lipid, derived lipid. Sorry, these are not derived lipid. They are termed as a conjugated lipid. Clear? They are the, conjugated lipid. Conjugated lipid. Right? So they can be of different type. For example, the lipoprotein can also be present. So different type are there. This is a one example phospholipid. Like this, we can be lipoprotein. Lipoprotein. That means lipid is having a protein group attached with it. Lipoprotein. Similarly, we are having sometime a carbohydrate group is also present in them, right? Sometime it is like some nucleic acid is attached to them. So accordingly, different name is given. Glycolipid is there. The chromolipids are there. Clearly. So glycolipid, and the fourth one is the chromolipid. Clear? So conjugated is done. Now let's talk about the derived lipid. The next one we have is a derived lipid. Now derived lipid do not have a glycerol and the fatty acid chain. Then what do these derived lipid have? So derived lipid, they are basically, their property is same. For example, the hydrophobicity is same. The hydrophobic nature they also have, but they do not have a fatty acyl chain. In this case, we have an example of the sterols, the cholesterols. You all know the fat is stored in the form of a cholesterol. Now this cholesterol, the steroids which are present, they are basically the 27. If you look at count the carbon, there are 27 carbon containing compounds. Clear? Now they are basically, when we talk about the various hormones like progesterone, the estrogen, they are the steroids. That means they're derived from cholesterol. So their structure somewhere or other look like a sterol. So this is the cholesterol. Clear, buddies? Hope it is clear till now. Okay. Now, moving on to the next, let's talk about the nucleic acids. So we have completed the carbohydrates, we have completed the lipid. Now let's talk about the nucleic acids. So nucleic acid, they are what? The example we have is DNA or RNA. Right? In a simplest way, the name is given as the polynucleotide chain. Polynucleotide chain. They are termed as polynucleotide chain. So that means different nucleotides are there, and nucleotides, they are joined together. So what are these nucleotides made up of? Nucleotide, they are made up of the sugar, the phosphate, plus you can say the base is present. Clear? Now, let's talk about the sugar. We'll be looking at all these nucleic acid in the chapter molecular basis of inheritance also, which is there in the class 12. But there are only few things which are related to this topic that definitely we'll be discussing. So, to talk about the sugar which is present in them. So, sugar, they can be riboses or they can be deoxyriboses. Riboses or deoxyriboses. Now, riboses, they are found in RNA and this is found in DNA. This is found in DNA and riboses, they are found in RNA. So, phosphate group is also present. Now, let's talk about the base. So, another name is written as base. So, let's talk about the base. So, base, again, we classify this into two different categories. For example, we have is a purine and second we have is pyrimidine. Purines and pyrimidines. So purines, they are what? A and G. Adenine and guanine, they are the purine A and G. Whereas when we talk about the C, the U and T. Clear? That is your cytosine, uracil and thymine. They are the pyrimidine, the cut. So out of these bases, when we talk about the DNA, in the DNA you are going to find the A, the G, the C and T. In the DNA you will find A, G, C, T. Whereas when we talk about the next one, that is a RNA, the bases which are present are the adenine, the guanine, the cytosine and uracil is there. The uracil is there, the DNA and the RNA. Clear, buddies? So this is how everything look like. So it is present in the DNA, RNA. They are the polynucleotide chain. The nucleotide which is present in the sugar, phosphate in the base. The sugars, I talked about the sugar also. Now one thing which you have to remember, there is another terminology that is termed as nucleoside. So what are these nucleoside? Nucleosides, they are what? Nucleoside is equal to base plus your sugar. So sugar can be riboses and deoxyriboses. They are termed as nucleoside. That means they have sided what? The phosphate group. So in another way we can write nucleotide is equal to nucleoside minus phosphate. Oh, sorry. Nucleoside equal to nucleoside is equal to nucleotide minus phosphate. Clear? Yes, buddies. Is it clear? Yes. So all the bases, they are present and the different you can say they are present in a, see, DNA is basically the polynucleotide chain and these polynucleotide chain, they are joined together. So these are the different structure of a adenine. For example, the your uracil and uracilic acid, I'll talk about it. What are these? So basically, buddies, let's talk about why these different names are being given. This is a direct diagram from the NCERT. So when we have is a adenine is what? Adenine is a purine, right? A and G both are purine. This is how a adenine look like. So two ring structure are present in the purine, whereas the pyrimidine only a single ring structure is present. So when we talk about this purine, suppose this purine is attached to this sugar. This is what? Sugar, ribose sugar, right? Which sugar is this? The ribose sugar, right? When it is attached to a ribose sugar, here you can say that this is adenine is present. This is adenine is present. So name will be derived it into the adenosine. Initially adenine, now it is a adenosine. So what do we call this? What's this? This is a nucleoside. So this is a nucleoside. Now when we talk about the nucleotide, in the nucleotide you can see a phosphate group is also attached. So this will come out to be the adenylic acid. Adenine, adenosine and adenylic acid. Here the uracil is there, which is a pyrimidine. Here the uridine will be there. This is how a uridine look like, because here the two OH structures are there. So two OH structure, that means this will be termed as a sugar. And this sugar is what? This is a ribose sugar. This is not a deoxyribose, this is a ribose sugar. And it will form a nucleotide. So nucleotide will be like this. We have a ribose, and here we have is a uridine uracil attached to it. Here we'll be having a phosphate group. So name will be written as uridylic acid. I'll write it here, uridylic acid. What will be the name? Uridylic acid, nucleic acid. Clear? Now when you look at this DNA, nature of the bond linking monomer is a polymer. So basically I told you this is a polymeric chain, polynucleotide chain is there. Now this is how a DNA or the DNA look like. Let's take an example of the DNA. Now the DNA look like, here they are made up of the polynucleotide chain. This is a polynucleotide chain, this one, and the another one is also a polynucleotide chain. Both of them, they join together, and in between them the hydrogen bond is present. That hydrogen bond which is present in them can be a double bond, can be a single bond, depending upon the type of bases which they are having. Now if we are going to focus this diagram, what you are going to see here, we have is, suppose in one chain we have is a thymine, on this another chain it will be attached to the adenine, and both of them, they are attached with the help of a double bond, and it is a double hydrogen bond which is present in them. Similarly, on the one chain, if it is guanine, it will be attached to the cytosine of another chain. The three triple bonds, they will be present. So they are reverse polarity. On one strand you will find a 5' to 3'. On the next strand you will find 3' to 5'. So basically they are not parallel, they are anti-parallel to each other. So the bond which is present in them, what is that? That is the hydrogen. So hydrogen bond keeps both of them together. So if we talk about the DNA backbone, see, they are the stacking structure present like this. So this is how the bases, they are present. Bases will be present like this. So this is a backbone. Backbone. So backbone is made up of sugar and phosphate. Sugar plus phosphate. Detailed structure we'll be looking in the class 12 standard, the molecular basis of inheritance in all the structures. Now, some basic things which you have to remember related to this is, these are the direct lines from the NCERT. 2 hydrogen bond is present between the AT. I told you in the AT you will find 2 hydrogen bond, whereas in the GC you are going to find the 3 hydrogen bond. Each strand appear like a helical staircase. It's like we have a wire, we have a imaginary axis, and on this imaginary axis these two strands, they are wound together. It is like this. Each step of ascent is responsible for the pair of bases. So here the pair of bases are present, right? It is like we have an imaginary axis, and around it they are being wound. Now at each step the strand turn 360 degree towards upper side. So it is like they are more going towards upper side. So they take a turn of around 36 degree. So in a 360 degree turn, total 10 amino acids or 10 base pairs are present. One full turn of the helical strand would involve the 10 step or the 10 base pair. Attempt drawing, the next one is pitch is 34 angstrom, right? The rise per base pair is 3.4 angstrom. The form of DNA which above mentioned, this is a classical example of a B-DNA majorly found in the human cell. Like this we have a A-DNA also, we have a Z-DNA also with us, right? Now moving on to the next topic, that is a protein. So majorly now we'll be talking about the proteins. So proteins are what? Proteins are the chain of amino acids, right? So they are the chain of amino acid. If someone say it is a homopolymer of amino acid, no. So proteins, they are what? That is the heteropolymer of amino acid. Polymer of amino acid. Heteropolymer of amino acid. So heteropolymer, that means different amino acids are there which are joined together. Amino acids, they are what? Amino acid, that means these are those which are having one amino group and acidic group. So here it is like a CH4 group, and we have modified the CH4 group. How? For example, this carbon, on one side it is attached to H, on one side it is attached to NH2, right? An amino group. On one side it is attached to, the carboxylic group, COOH, right? Here the differential or the variable R group is present. The carbon which is present in the center, that is a chiral carbon. Now this R is H. This R is H only in case of simplest amino acid, which is a glycine. So R is equal to H in glycine. Whereas in various, you can say the type of amino acid, they are variable R group is present. This R group is variable. If someone asks you how many amino acids are present, more than 200 amino acids are present. But there are only 20 amino acids which helps in the formation of protein. Yes, 20 amino acids are there, and these 20 amino acids, they help in formation of protein. Right? Today we have discovered 21 and the 22nd amino acid also. So 20 amino acids, they are 21st amino acid we have is a solanocysteine. Yes, it is a modified form of cysteine. Cysteine, you know, it's an amino acid. So like this we have is a solanocysteine is present. Clear? And one more amino acid is also present. I don't remember the name, right? Now these, I'll be talking about the basic properties later on, but first of all let's talk about the types of amino acid. So amino acid, they can be non-polar, right? They are aliphatic. Aliphatic R group are present. For example, if H is there, that means on the alpha carbon, so it will not be a chiral carbon because two H group are there. It will be termed as a glycine. If it is having a simple CH3 group present, it will be termed as a alanine. And the valine also, it will look like this. So this is what you guys have to remember. Don't go detail into the structure. Don't go detail into the structure. Like these are some basic things which you have to remember. We have as a leucine, some modified R group. Now the methionine is also there. Now the methionine is having S group with them. So what is this S group? S is what? Guys, S is what? This is a sulfur present. Then we have as isoleucine, aromatic ring. For example, phenylalanine, tyrosine and tryptophan is there. And if we have a polar uncharged group, serine, threonine, cysteine, proline, asparagine and glutamine are present. Clear? Now moving on to the next, we have other positively charged amino acids also. For example, lysine, arginine and histidine. We have a negatively charged amino acid group are also present because they are COO negative group present, means extra. Otherwise it has COO negative group. Aspartic acid we have is a glutamic acid is also present. Clear? Now let's talk about this amino acid. Amino acid can exist as a Zwitter ion depending upon the pH. Right, basically at a specific pH it can form a Zwitter ion. That means both of them, they will be present in the ionic form, NH3 positive and CO negative. That is a pKa value, so that they are termed as a Zwitter ion. So they can exist as a Zwitter ions. Now let's talk about the protein. So proteins, they are polypeptide. So polypeptide, that means different amino acid they join together, what they form is a polypeptide. So you should know 20 type of amino acids are there which helps the formation of protein. Otherwise there are more. They are the heteropolymers and they are not homopolymer. Clear? Now structure of protein. So to talk about the structure of protein, one second. Right. So to talk about the structure of protein, so we have a primary structure of protein. We have a primary structure. So primary structure is what? Primary structure is a linear structure of the amino acid. Clear? So it is having a linear structure where on one side we are having a see, it is like a linear structure. Linear structure. So this linear structure is termed as your primary structure. I can show you this also. So this let me show you. I'll write it here. This is how a N terminal, this is a C terminal is present. It's a linear structure. Let's talk about the secondary structure. Let's talk about the, it can show the secondary structure. For example, the beta plated and alpha helix, if they form a helical structure like this. Now the third one we have is a tertiary structure. tertiary structure. So tertiary structures, they are, guys, what? Tertiary structure. That means these are those structure which forms a rounded structure. Like mean one polypeptide chain has been rounded like this, and they form a amine. They form a active site, right? So this is, for example, your myoglobin. Myoglobin is very good example of this. Now the last but not the least we have is the quaternary structure. Quaternary structure. So quaternary structure, buddies, they are what? For example, different polypeptide chains are there, and these are joined together with a iron group. Let's take an example. They are joined together, and for example, the example we have is hemoglobin. We have is a hemoglobin, myoglobin and the hemoglobin. Many times this was all like hemoglobin is what? So this is how a primary structure look like. So this is a linear chain of the DNA. Now here you can say this is a secondary structure, which is a alpha helix, and this is a tertiary structure where they have formed a structure, they form a ball-like structure like this. Now, some basic properties, you all know the collagen, the trypsin, and the insulin, the antibody receptor, the GLUT1. What are these? These are your these are your proteins also, and they are protein only, and they form a various different kind of functions in our body. Now, moving on to the next part of this chapter, that is the enzymes. So, enzymes, they are what? Enzymes, they are basically made up of proteins. Right? We have an exception, ribonuclease, RNA enzyme. And name of this RNA enzyme is ribonucleases. Right? Now, to talk about the enzyme, they are having active site. Right? Now they are having active site. So this is that site which bind to substrate. In a normal way also the reaction can occur, but enzyme do what? Enzyme decreases activation energy. Activation energy. So activation energy is that energy which a substrate has to cross, or it has to match with the energy of the product. So this this reduction in the activation energy, this is done by enzyme. So we have different category of these enzymes also. We'll be talking about it. Now let's have a look. Right? Now let's talk about how does this enzyme function. Now these enzyme, how do they do the conversion? To understand this, you have to know that each of these enzyme, they are having a active site. So enzyme, it binds to the substrate, it forms the intermediate, and ultimately a product plus enzyme we are going to get back. Clear? So this substrate bind to the enzyme active site, intermediate, that is the substrate enzyme complex is formed, and later on it gives a product. Clear? Now this is all about the activation energy. Now see, this is a substrate and this is a product, right? And here we have is a potential energy. So if we have to form a product, now this is a barrier which the substrate has to cross. This is a potential energy barrier. So this is a activation energy. This is without enzyme. So when we use this enzyme, enzyme decreases the activation energy, and when it decreases the activation energy, the substrate is converted into product, and enzymes, they are released as such. Now there are various factors which affect the enzyme activity. For example, we have is a pH, we have is a temperature, we have is other substrate concentration also. Now listen to this very carefully. Now when we talk about the enzymes, they are made up of proteins, and we know whenever we increase the temperature, the proteins, they denature. We have an exception. For example, when we talk about the Wnt polymerases or other polymerases, the Taq polymerases, they remain active at high temperature, but they do not remain active at high temperature. So temperature, the pH, and the substrate concentration, all of this, they affect the enzymatic activity. First of all, let's talk about the temperature and the pH. Now, enzymes generally function in the narrow range of temperature and the pH. Now, each enzyme show a highest activity at particular temperature in the pH, which is termed as its optical temperature, optimum pH. Clear? Now, activity decline both below and above the optimum value. So that means if particular temperature is not provided, what will happen? If that is not provided, what will happen? The the enzyme will not be able to function because they are made up of proteins only. Low temperature preserves the enzyme in a temporarily inactive state, whereas a high temperature destroys it denatures the enzyme. Now, this is all about it. Now, pH, that means the activity is specific or the highest at a particular pH. You can see that. Similarly, at a particular temperature, you can see the activity is like this. Right? Now, let's talk about the third important factor, which is a substrate. What is the substrate concentration? This is a substrate is given. This is a velocity of reaction. You know, initially, whenever we increase the substrate concentration, the velocity of reaction increases. But there comes a particular time, that is a Vmax. This is a maximum velocity by which a product is being formed. That is up to a particular point, because now at this point what has happened is all the enzyme, they are saturated with substrate. They are saturated with substrate. They are saturated with substrate. Clear? Are you getting this point? They are saturated with substrate. Now we have to understand another terminology, and what is that? That is the Km and the Vmax. Now Km is what? Many times this question is being asked in the examination. What is a Km? Km is that substrate concentration at which the velocity of reaction is half. This is the Km value. Now let's talk about the inhibitor. Now we'll be talking about basically those inhibitor which affects the reaction, overall reaction. Now here the inhibitor, when it closely resembles the substrate in their molecular structure, this is termed as a competitive inhibitor. So that means inhibitor will do what? Inhibitor will bind to active site of enzyme. Active site of enzyme, and when it bind to active site of enzyme, what will happen? It will block that particular enzyme. See, what has happened is whenever the substrate is there and the substrate and this enzyme, they closely resemble each other. When they resemble each other, instead of binding to the substrate, the inhibitor bind to the active site of enzyme and it blocks the activity. This is termed as a competitive inhibitor. Clear? So due to the close structure similarity, the inhibitor competes with the substrate for the substrate binding of the enzyme. In this case, a very beautiful example is the inhibition of the succinic dehydrogenase. This is an enzyme which is inhibited by the malonate, and it resembles the substrate which is succinate. If you look at the structure of the succinate and the malonate, they are very much similar to each other. And whenever this malonate binds to the succinic dehydrogenase, the product is not being formed. So this is termed as the competitive inhibitor, and this competitive inhibitor is found to control the bacterial pathogens. Clear? Right? Clear? Similarly, we have is a non-competitive inhibitors also, right? I'll talk to you about the non-competitive inhibitor also. So non-competitive inhibition. Non-competitive inhibitions are those inhibition whenever the substrate, in this case the substrate binds to enzyme, right? Substrate binds to enzyme. Similarly, inhibitor also binds to the enzyme, but inhibitor do not bind to the active site of that enzyme. If it does not bind to active site of that enzyme, now what will happen? The enzyme activity will not be blocked. And if enzyme activity is not enzyme active site is not blocked, that means now the substrate can bind. Are you getting this point? So this is termed as a non-competitive inhibition. The cyanide, this is a very good example of the non-competitive inhibition. Now where there are various classification of these enzymes, let's have a look. For example, those which help in undergoing the oxidation and the reduction, that is termed as the oxidoreductase enzyme. Transferase are those which help in the transfer of a particular group. For example, any group other than the hydrogen. So this will be termed as a transferase. Hydrolases are those enzymes which causes the hydrolysis of the ester, ether, peptide, glycosidic linkage. They will be termed as a hydrolases. Next we have is a lyase. Lyase does what? It removes the group from the substrate. Lyases. Isomerases, they are what? They are the interconversion of the optical isomers, right? Optical geometrical isomers, whatever it is, they are just converting it. Ligases are those ligases, they are also termed as a feviquick. So they joins the two molecules together. Ligases. Clear? So what all things we have discussed today? We have talked about your proteins, we have talked about the carbohydrate basic structure, the lipid basic structure, we have talked about the enzyme, we have talked about the nucleic acid. All these things we have completed, right? So we have talked about the lipid, the carbohydrates, the proteins, and the very important part of this chapter, that is the enzyme. Now these enzyme, the inhibition, the competitive and non-competitive inhibition, they also affects the Km value. How do they affect the Km value? That we'll see in the detailed session. We have to learn some basic structure of monosaccharides also, for example the glucose, the fructose, etc. So that also we'll be discussing in the detailed discussion. So let's wind up the session over here. This was just a summary session to give you an overview of this chapter, that what all things are included. And you should have an idea about the biomolecules which are present inside the human body. So let's wind up the over here. We'll be meeting you next. Till then, take care, keep studying, keep yourself safe in this time. Bye-bye everyone.